Okay, question 1 is a knowledge-based question. So it says, draw one line from each substance to the correct site of production. Well, bile is made in the liver, amylase is made in the salivary gland, and protease is made in the stomach. Each of those is worth 1 mark. Okay, figure 1 shows the proportions of fat and protein in the meal. After the man eats a meal, the percentage of amino acids in his bloodstream increases. Give 1 reason why. What you should know is that protein gets digested or broken down into amino acids. And because of that, when you eat the meal, you're going to get an increased amount of amino acids. So you've got to look at these results and decide what 2 tests are in the sample. Well, we can see from this table that you get a positive result for Benedict's and a positive result for iodine. Well, Benedict's tests for glucose, so we should have ticked glucose, and iodine tests for starch. Both of those are needed for 1 mark. You then had to complete the table by putting in the test, the positive test for iodine. Well, it goes blue stroke black. Question 2.1. Name 2 cell structures that are in eukaryotic cells but not in prokaryotic cell. The most obvious one of these is a nucleus, because nucleus are only found in eukaryotic cells. The other ones that you could have had were mitochondria, you could have had ribosomes, vacuole, or chloroplasts. An electron microscope has a higher magnification than a light microscope. This shows you what a prokaryotic cell would look like under a light microscope. You've got to say one other structure that you'd see if it was an electron microscope. Well, you are most likely to see a plasmid, and just for your information, a plasmid is a circular bit of DNA, something like that. Question 2.3 is a tricky maths question. You've got to convert this into standard form, but interestingly, all the standard form answers are written in metres. For the first thing we have to do is to convert 0.006 millimetres into metres. We do that by timesing by 1,000. If I times that by 1,000, I get this. And then to convert that into standard form, I then have to work out how many places I need to move the point. So 1, 2, 3, 4, 5, 6. So that's going to give me 6 times 10 to the minus 6 metres. So question 3 is about a disease called cellulitis. Important things, it's caused by bacteria. So complete the sentences, use words from the box. The bacteria that causes cellulitis releases toxins, which cause a rash on the skin. Cellulitis can be cured by taking antibiotics because it is caused by a bacteria. 1 mark for each of those answers. When a person is infected with cellulitis bacteria, the white blood cells engulf and destroy bacteria. What is this called? This is called phagocytosis. Okay, in the first graph it shows you the antibody production after the first infection. You have to show what would happen if you got infected again. Well, we know if you get infected again, then antibodies are produced more quickly and in larger quantities. So you're looking for a steeper rise in your graph for the mark. Experiment, it's asking you what you're going to use to control the temperature of the mixture. Well, we control the temperature by using a water bath. You then have to draw a line of best fit on this graph. Remember, lines do not have to be straight. So on this graph, what you're looking for is a nice curve of best fit, something like that. To answer 4.3, the starch suspension contained 0.8 grams of starch. Calculate the mean rate of starch digested at 33 degrees C. You've got to go back to the graph and work out how quickly starch is digested at 33 degrees C. On my line of best fit, it takes 3.2 minutes to digest the starch, so I'm going to do 0.8 divided by 3.2, and that gives me the answer of 0.25. Your answer might differ slightly because it depends on your line of best fit. Now, 2 ways that you can improve the precision of the data. You could do smaller temperature ranges. At the moment it's done every 10 degrees. You could do it every 5 degrees. So you could do 5, 10, 15, 20, etc. And at the moment the solution is tested every 30 seconds. You could test the solution to see whether the starch is broken down maybe every 10 seconds instead, to have smaller time intervals. This one, what is meant by the term accuracy? Accuracy means that measurements that are close to the true value, that's worth 1 mark. Okay, in this question we're looking at how things move into the gill. So in the first picture you've got a high concentration of oxygen outside, the oxygen is going to be moving in, so we're going high to low, so that's going to be diffusion. This one, we've got a low concentration on the outside, a high concentration on the inside, so it's going to low to high, and therefore that is going to be active transport. Each of those for 1 mark. Now the last question in your paper doesn't appear here. It's about the adaptations of the alveoli. So how is the alveoli adapted? There's lots of alveoli, that's worth 1 mark. That means that there is a large surface area for gas exchange. That's your next mark. Then you could say there are thin walls in the alveoli, which is worth 1 mark. The reason that's important is because the gases do not have far to move. So that's could be worth a mark. Another one that you could have is that they've got a good blood supply for 1 mark. This helps maintain the concentration gradient, again for a mark. So what they're looking for, if I just finish writing this, you get 1 mark for the features, so lots of alveoli, or the alveoli got thin walls, or they got a blood supply. So any 2 of those for 2 marks, and then for each one you have to give the explanation in order to get the full 4 marks. I hope you found this useful.